Hello everyone. Today we will be looking at the demonstration and tutorial video of spectrophotometer. Spectroscopy is a commonly utilized study that quantifies the interaction between the light and the sample of interest. Spectrophotometer is a device that measures the intensity of light as a beam of light passes through the sample solution. So, a light is an element that can be reflected, scattered, transmitted, or absorbed. This instrument is a combination of spectrometer, which give rise to a light of any wavelength, and a photometer, which measures the light intensity. Spectrophotometer is broadly used in analytical chemistry, and it is used for the quantitative analysis of analyte. This is usually done if you want to find the concentration of an unknown sample, the rate of the reaction, growth of microorganism, etc. Spectrophotometer optically determines the absorbance or transmission of characteristic wavelength of light by a chemical species in a solution. Here, each molecule in the solution absorbs light at a certain wavelength and is unique in the spectral pattern because of the number and the arrangement of the characteristic functional group present inside them. So, the relation between the amount of the light absorbed and the concentration is usually established through Beer-Lambert law, which is basically stated as A is equals to epsilon BC. The Beer-Lambert law states that the quantity of light absorbed by a substance is directly proportional to the concentration of the substance, which is the C, which is represented as C here, and the path length of the light through the solution. Here it is represented through B, and A is the absorbance, and epsilon is the molar absorbability of the solution. Uh, if we look at the instrumental part of this, then the instrumental part of spectrophotometer consists of a light source, a digital display for the output, a monochromator, a wavelength sector to transmit a selected wavelength, a collimator for straight light beam transmission, and a photoelectric detector, a cuvette in which is used to place the sample. So the working is in a manner that the sample which is of our interest is placed in the spectrophotometer. The light source here, it is used to shine the sample, and the monochromator placed, it splits the light into each colors or into individual wavelength. So the light's wavelength that hits the sample, which is held in the cuvette. Now we will see how these cuvette looks like in the demonstration video, and I can give you that information that these cuvettes are available in as plastic, glass, or quartz cuvette, and we will be requiring a very careful handling with these cuvette, as even if there's a slight existence of fingerprint, they can actually have the capacity to alter the results. So the light that passes through the sample is basically read and interpreted, which is being displayed at the output. Let's now move to the demonstration part of the spectrophotometer for better understanding. We will now look at the demonstration part of UV visible spectrophotometer. In this video, we will do a protein analysis through the spectrophotometer, and this will help you for better understanding of the instrument as well as for the sample testing. So the instrument that is placed right in this video is a UV visible double beam spectrophotometer from Lab India. So as we have already discussed the principle of spectrophotometer in the tutorial video, what is different here is the terminology double beam spectrophotometer. So here what happens is the single light beam in the double beam photometer basically gets splitted into two beams for the diffraction process, and here the monochromator, which was shown in the tutorial video, play its role by selecting the specific wavelength of light
for spectrometry process. So throughout this measurement, what we see is here the measurements are done with a reference point that is set into the spectrophotometer for measurement process, and similarly later on analysis. So if you look clearly at the instrument has a power button which is placed right behind this instrument. It is a power switch button, and the instrument also has a few of the parameters. So we can actually operate this instrument directly here, or we can connect it to the desktop through a USB port, and we can operate the entire process in the system. So if you look in the instrument, there are few modes which you can have, which you can operate from the display, if you from the keys over here. So there are two lamp which is, as it is a UV visible, so we have a UV lamp as well as a visible lamp. If the lamp is in the process and if it is working, there will be a symbol of it to be enlightened, and in the other way it won't be. So these are the few like basic mode, the quantitative mode. For example, if I'll go to the system utility, you can you can you can actually look at your lamp, and you can, for example, this is the lamp service. You can switch on or off your D2 or the other visible lamp from this system itself. So as I said, the process can be done from the instrument as well as from the system which we will say. If you look at the how does the sample cabinet looks like. So as I said, it is a double beam spectrophotometer, and all the analysis which takes place is always done by keeping a reference sample or a control sample throughout the experiment. So this is the slot for the control sample, and the light always comes here in this direction. This direction and the placement of cuvette is important, that we'll discuss in the future. And the other cuvette which is placed in this cabinet, and the light enters from this side. So that's why we have two beams over here, and that's why the name suggests the double beam spectrophotometer. We will now look at the protein estimation, and this estimation we will do through Bradford method. Bradford method is the most commonly and widely used method for protein estimation. So the protein estimation method, this method is based on the principle that the protein binds to the dye, which is a Comazi brilliant blue, in acidic solution, and it forms a complex whose lambda max, or the maximum wavelength, is around 595. Whereas if you look at the free dye, then it has a maximum wavelength of 465 nanometer. The intensity of absorption of protein dye complex is proportional to the concentration of protein. So the dye strongly binds to the positive charge group of protein, and also to hydrophobic regions in the protein. So as a result, there occurs a formation of blue color in the sample, which has a maximum wavelength of which is around 595. So for the experimental process, we have taken a stock solution of protein, and we have serially diluted it in 3 different volumes, which is having an increasing amount of protein, and we have made this final volume to around 500 microliter. And now we will be adding the reagent Bradford inside it, so that we can do the protein estimation further. We will start the experiment by doing a blank calibration. It is always advisable to use blank as a material in which your protein or the sample is having a solvent. Basically, it is always advisable to use blank for something which is a solvent in your sample. So here, since we have dissolved our protein in distilled water, so we have kept our blank as distilled water. And as I said, since the system always works by keeping a reference sample, so we will put, these are the cuvettes. Now cuvettes are basically the transparent vessel in which the analysis takes place. So these cuvettes are made up of quartz, plastic and glass. So this one which I am using right now is a quartz crystal, which is always can be used in the UV or visible range. However, if you are using a plastic cuvette, so it is always advisable to not use it during the UV scan. So we will do the scanning at 595. So either the quartz or the plastic will work fine for us. Now for putting the sample, we will put the two, initially we will put the two samples. So since I said that the light in this slot comes in this direction, so if you look at the cuvette clearly, the cuvette has two surfaces. One of the surface is very transparent, however the other one is a little opaque one. So we want the light to pass through it, so we'll keep the transparent surface in this direction because the light is entering in this direction. Similarly, the second cuvette initially, which will in the later section will become the sample cuvette, will be placed, and if you look at this cuvette also, so this cuvette also has a transparent surface and a little opaque surface. Yes, so now similarly, as I said, in this slot the light comes in this direction. So as I said in my tutorial video also, we should avoid any kind of dirt or fingerprint on the transparent side from where the light is entering. So before placing the cuvette, we will just wipe the transparent part of the cuvette and then we'll place it inside. So right now we have two cuvette of our distilled water, which is our blank sample. So now we will go to the basic mode, and we want it to the sample which we have kept is our blank, so we'll do a blanking here. And you'll see that the reading comes as a reading of a blank sample. So this is our first dilution, and I have added Bradford solution inside it. So as I said in my previous in the previous part, that there occurs a development of blue color. So always try to expose the color solution from light, and now we will take the initial reading of it, as previously we have already calibrated our system. And now, as I said, this one is our sample slot now, and we'll put the sample over here. And if you see that there is an observed reading which is 0.274. So this value that is being displayed here correspond to the protein content which is present in our sample. Let's now look at the sample, the other dilution which we have made. So this is the second solution, this is the second sample, and I will again place it in the slot. So if you see the reading, this is again the respective protein content. So as I said that the dilution was in the increasing amount, so that can again be reflected in the readings over here. And this is our final solution. So I have added Bradford here. Make sure that you avoid any kind of bubble because this is going to affect the reading also. So this is the third sample, and I'll wipe it to avoid any fingerprints over here, and I'll place it here. So I guess throughout the process it's quite obvious that you have to close the lid to make that darkness over here. And if you see, this is our third sample, which yeah, this is our third sample, and again, if you see the readings is somehow similar to the second one, which suggests that the protein content hasn't varied much in the third sample. This is for the spectrophotometer demonstration part, and as I said, the instrument can always be handled through a desktop, so you can handle it locally, or you can connect a USB port, and then you can take the readings in the desktop also. Thank you.